Well, shall we start or no, we wait?
Sí.
Well, good afternoon, everybody. It is a pleasure for me to introduce you Jesus Marco. Jesus studied in the University, or did a PhD at least. Did you study also in the University of Cantabria? He did the thesis in experimental particle physics, working at the Delphi experiment in the CERN, where he was one of the coordinators of the data analysis in search of the Higgs boson. He subsequently worked in the CMS experiment on the development of distributed computing system, the grid systems, and in parallel on several European computing projects, coordinating the interactive European grid project and initially the Deep Hybrid Data Cloud project. He was also responsible for the installation of the Altamira supercomputer node in the Institute of Physics in Cantabria, and he was also director there from 2004 to 2007. He was coordinator of the CSIC area of physical science and technology, and more recently he was vice president for research. He was contributing to the creation of the interdisciplinary thematic platforms that some of you already know, the CSIC connections, and the preparation of the CSIC 2030 challenges white papers. He's currently working in advanced computing and e-science group at the IFCA, exploring ideas to be applied in data science coming from neuroscience. So thank you very much, Jesus, for being here.
Thank you very much, Claudio. Thank you very much for the invitation. It's really a pleasure for me to be here in this center that I appreciate so much. Although I have not been here so many times, but maybe this will change in the future. And thank you very much for being here today. It's an unusual time for me, but apparently for you it's very usual, so that's nice. And well, my talk is going to cover some of the points that I have written in a preprint in arXiv. The title is exactly that: implementing engrams from a machine learning perspective, machine for prediction. It's also very nice to see so many young people around, because all the young people in my center has criticized very strongly my preprint. They say, oh, you should write something that really makes sense. So you will see that part of it makes sense, but not. I will start with a very brief introduction. Why I'm talking about engrams? Well, if you take volume 5, Brain, Mind, and Behavior of these white books, and you may find there the word engram written somewhere, in particular due to the referring to this paper of Josling and Tonegawa, because not so many people talk about engrams any longer. And engrams are in principle the way that the memory is written in our brains in some way. And when I returned to my center 1 year ago, more or less, I thought about memory, basically because when you get older, you start to be more and more and more worried about memory. And then I thought that we never talk about memory in computing, because memory is something that is more or less trivial, no? While it is a big problem with GPUs, because you have to move the data all the time, no? And that's complex. So I started to read about engrams. And first thing I learned, and that you know likely much more than me about all these things, in particular because you are in a center that is devoted to complex systems, no? That neural circuits are really complex. And you may know the Blue Brain project, that is one of the, thank you, is one of the projects that was launched already more than 10 years ago, and that was supported heavily by European fundings to try to make the connection between the microcircuitry in our brain and how does it work from anatomy, let's say, to the functioning. Well, you may also know that just a couple of years ago, Google has published the connectome of the brain. This is really only 1 millimeter, you see here, and it's really, really, really complex. And even more, you see here in red the important role of connections that have the inhibitory connections that already, let's say, 10 years ago were not so considered so important. And as someone told me, that was one of the points, of the few points that Cajal missed at the time. Cajal really did an incredible work understanding all the structures in the brain, but this is one of the points that he did not explore so much. And the Blue Brain project is right now checking things like metabolic state machine as an important structure in the brain. So what's the message? The message is, yes, it is very complex. And the second message is, it is so complex that now we are really going to the, not only to the connectome, but also to the internal working of the cells. Okay, and that's, to my knowledge, is a problem that is not solved. In fact, we have another white book on the cell and the origin of life. And if you go there, you realize that, of course, cells are still a mystery. So what about structures in the human brain? This you already know also, but there are some points that I was not aware and that are interesting. First one is that a neuron, a single cell, may have, well, 10, 20, it is widely varying dendrites, and usually one axon. And of course, the length is not the same. And this image that you see here, okay, that likely is the one you have in your heads, is not true at all. It's not true that really the axon connects to the synaptic terminal of the next neuron, that is a dendrite or dendrite. And what happens in real life, in principle, and I say in principle because establishing there's real synaptic connections is not easy, is that this axon is kind of an en passant connection that each 2 microns make a connection with another neuron or several connections with the same neuron. Okay. So this is the first thing that I didn't know. That is in this nice review of Sudov of a couple of years ago, the cell biology of synapse formation. The second point is that there is a canonical release machinery for the connection from the axon to the dendrite. And this release machinery of the neurotransmitter is a standard, but each response to the neurotransmitter is specific in each dendrite. So really, it's really, really complex, you know, likely, no? That is a very complex mechanism. And one of the most worrying things is that we sometimes forget that this is a critical circuit, that the neurons, even in pyramidal, that is only one kind of neurons, are mediated by interneurons, and the role of these interneurons that are usually inhibitory is also not well understood. And as I said before, I'm not an expert, and I will not talk about, I will not tell you what is the role. I will only telling you that all this complex system deserves more attention and more interest to understand the potential role of these connections. Another important point that I learned in this previous paper of Sudov is the evolution of the neural plasticity. So along the prenatal period, we are positioning the neurons, so they are created and positioned, while in the next 2 years of postnatal, there's exuberant synapses growing in our brains. So it's quite important that these 2 first years of childhood go properly, okay? And it's well known that effects on that 2 first years may affect the rest of the life. But it's also true that after that, there's a period of around 20 years of pruning, where around 40% of these previous connections are established. And then after, basically, when you are 20 years old, well, all people smile because we know what happens. It's not completely written here, I think. And this is described in this scheme, okay, nascent synapses, mature synapses. And then these mature synapses, another thing that I didn't know, is that many of these synapses are established and it's not possible to change them much more. And some people say that this explains why old people do not change opinions, okay? Well, and then there comes the sad question when synapses are eliminated, and then this starts the degenerative problems, and this kind of problems likely, nobody knows. So plasticity makes our brain 40% of spines in pyramidal neurons in sensory and motor cortex are replaced every 5 days. Only 60% are stable. Okay, so this is likely something that we don't think about, that we may think that our connections are stable, but it's not true for a large percentage. But even more striking is that the hippocampus, that as I will say later, it may be a central node, 100% of spines turn over every 2 weeks. So this means that there's some mechanism that allow us to consolidate our personality in our hippocampus if this hasn't place there. But even if 100% of spines turn over every 2 weeks, okay? And then another question, why do synapses form or disappear? I have said that the axon goes this way. So let's say you are in the brain, you are a neuron, and then you send the axon there and it tries to connect. Why does it connect to somewhere to a neuron or to another neuron? Okay. It is known that there's non-physiological promiscuity, and there's some role of something called G protein couple receptors that I don't know. And then there's the glia, the supporting material in the brain, with astrocytes information that may give tripartite synapses, microglia in eliminating connections. All these are mysteries that are being studied. Even more, a large mystery is what intracellular signals induce synapses. But this is not only true for neurons, it's true for all the biology, for all the cells. The intracellular signals are really a hot topic in these days. I hope you know this, no? And maybe you know all I'm saying, no? Or maybe I'm not saying true things, you forgot. As I said before, don't be too confident on what I'm saying. Eh? Well, now the point why I'm interested in this topic, or why I think many of the people in computing are interested in these topics, and they don't say the real reason is, okay, everybody wants to understand how the brain works. But everybody knows if they say that they want to understand how the brain works, they will be automatically disqualified for science, okay? Because this is well known that you cannot get an answer. So what's the trick? The trick is you say, okay, well, the brain is so powerful with so efficient that we want to understand how the brain may work, and the ideas can then go to computing. And as computing is so intensive in energy, we may find a way, and that's a good reason to understand why how the brain works, okay? So I have here four interesting papers. One is quite interesting, is towards next generation artificial intelligence, catalyzing the neuro AI revolution. This is a paper of October of last year, signed by many of the popes in the area. And that's one of the reasons that I'm trying to push forward on this direction. And I would be very much interested in setting up a kind of master or similar initiative to promote this interaction between neuroscience and computing science. Because even if there are a lot of connections, the experts are still quite apart, as you see in the literature, what they publish. So another important point is that neuroscience is using more and more deep learning tools, and sometimes they don't know how to use them. So it's the same neuroscience that provides the ideas that is using then the tools to understand their data. Then there's these other ideas in neuroscience of computational theory, in particular regarding hippocampus. And then there's these other large reviews from brain science to artificial intelligence. This is also relatively recent, it's 2020. So you see that the area is quite hot, but quite dispersed also. Right? Well, so I was there 1 year ago, and I said, okay, what can I do to understand something? And then to understand something, I took this example that maybe all of you know, maybe know. Who of you don't know about the Jennifer Aniston neuron? Okay, so the others know, so that's a good point. So Jennifer Aniston neuron. I read this in this book, a very interesting book from Juan Lerma and José Luis Rozas, El cerebro y las enfermedades del alma. And the point is this point, a bit of history and story. I will start with the story. Maybe I will not be completely correct, but the idea is that, and this is interesting because already in the 60s, there was a joke in the community that the 60s, their knowledge about the brain was not so good. And at that time, the theories of Freud were also quite popular. And so, of course, the influence of your mother on your life was so strong, was considered so strong, that somebody said, okay, and if you have your mother in a neuron in your brain and you remove this neuron, what happens? You remove all your problems with your mother, no? Well, this was not the case of the mother, but it became the grandmother fell. So you have all the information about your grandmother in a neuron, and if you remove the neuron, then you forget about your grandmother, okay? In fact, there was a doctor in Poland that was working in the Soviet Union with patients, and that has already explored the possibility of removing neurons from patients, because this was also at times, and that he, I don't know if the name is around, but he was already propagating this idea that you may have a concept in a neuron, and if you remove this neuron, you miss the concept in your mind, okay? The surprise was, after all this story about grandmothers, that in an experiment in 2005, and it's not exactly an experiment, there's only one possibility that researchers have to see what happens in a neuron in the brain of the people. And it's when you are an epileptic patient and you have so strong epileptic problems that you have to remove part of your brain to cure these epileptic accidents, okay? We have, by the way, we have researchers in CSC that work with doctors in Madrid to exactly determine what section has to be removed for. So the idea is that you have an electrode and you test with the electrode the regions, okay? So while doing experiments in 2005 with patients that were being studied because of these epilepsy problems, Rodrigo Quiroga and colleagues found that when some patients were presented with an image of a famous actress at that time, Jennifer Aniston, there was a neuron that was spiking, and when they presented images of other actresses, this neuron did not spike, okay? But it was not only Jennifer Aniston, it was also Jackie Chan. And it was not only one image specifically of Jackie Chan, but any image of Jackie Chan. And it was not only the image of Jackie Chan, also the text, a text written Jackie Chan, or an audio where Jackie Chan word was said, okay? And it was only a neuron. So apparently in your brains, you have a neuron where Jackie Chan is coded, for those of you who know Jackie Chan, okay? Well, this, I asked Juan Lerma, come on, this is true, or this is just, what does the community say about this, no? So he told me, yeah, it's true, it's true. Yes. Yes. It depends on what you think it's a concept. Yeah. Okay, I checked what the community said, and I found very interesting paper for 2019 that said the value of failure in science, the story of grandmother fails in neuroscience, saying this was not true, this was an error, and the value of failure in science is that we learn from that we have. Okay, so there are a lot of people that think, and this was also one person from the Department of History and Philosophy of Science, okay? But it was replied immediately by other people saying, no, no, this is working. So community does not know really if this is true or it's not true. In fact, just this month in May, Rodrigo Quiroga has published a new paper, an integrative view of human hippocampal function, differences with other species, and capacity considerations. Forget about differences with other species, because this is another topic, because he thinks that humans are so special that we only have this possibility to associate a neuron to a person or to a concept, okay? And what he says in that paper is that he's still confident on this because it has been tested with many, many other examples, including concepts like banana. Maybe banana is not a concept. It's more closer to a concept than Jennifer Aniston, okay? And what he also says there is that also what has been checked is that when you learn about this actor that I remember, Brolin, okay, and you know about image associated to Brolin, and the people are they are shown, they see both images and associate them together, then this gives also an association between both neurons. Okay, so now the neuron of this actor will spike when the people sees the Eiffel Tower and vice versa. So there's an association. So it's not only that there's a neuron for a concept, but there's also association between the neurons. Okay, so he even goes further in that direction. Okay, so I thought, okay, the community is divided, so how to address this? Okay, how to address this? What would you do? Well, we can check experimental evidence. For me, the first one is experiments are with animals. So understanding the circuits from worms, and you know that C. elegans is the first, not the only, is the first animal that was completely known all the circuits for the neural working, okay? To primates, the electrical response, the electrodes measuring the activity of a neuron. There's a lot of papers about that. Functional magnetic resonance imaging, okay? Markers of the genetic of synaptic connections, the clinical evidences of problems associated with brain lesions, okay? That in fact is part of the starting of the story, no? There are many problems when you start to check all these techniques. The first one is that, for example, for one of the, let's say the most obvious ones that is measuring the electrical response of neurons, is that simultaneous measurements of activity of multiple neurons for sufficient time, there are very, very few, okay, and not in a human brain. In fact, the last years the situation has improved a lot, but still there are many problems there. If you can check an article of a couple of months ago from Oscar Herreras from Cajal Center, saying, for example, that much of the theta and delta oscillations that we see that are in fact global fields are being interpreted as local fields in many cases, and this is not the case. The way that the electrical signals work in our brain is really not well known. And this is a very fundamental problem that we can discuss maybe later. The fact if these electrical patterns are really the key point, or if they are just the signal of the activity at the cellular molecular level. It's not obvious. The knowledge of the connectome, of course, I have shown you the previous image from Google, but even in this C. elegans, it's not obvious how to understand this. I found only a couple of papers describing special effects on the C. elegans when you touch it, how it moves, and they understand, and this is completely chemical circuits, molecular circuits, let's say. The interpretation of animal experiments, that's another important problem. They are starting in these last years to try a protocol to categorize the animal experiments. The mixing of multiple models and problems. One of the first problems I found is the role of the hippocampus and the spatial neurons. So you know that there was this Nobel Prize a few years ago about the structure, a grid structure on the hippocampus cells to reflect the space. This is now has been extended to other type of spatial cells in our hippocampus, in principle, also even to temporal cells, and the community is discussing all the time about this. But this is much more difficult even than the previous problem. And then of course, the previous issues that the researchers know that in principle, we have a limited ability to memorize information in neurons, in the dendritic connections, and also limited ability to recall memorize information. So when we address this problem, we have to have this in mind. Our brain is limited. It's not without limit. No? Regarding concepts, situation advanced a lot because the functional, as you may also know, with functional imaging of magnetic resonance, the progress in the last years increasing the field of the Teslas and making more and more maps. You may find examples like this one from the Gallant lab, where they have a map of the brain reflecting the concepts that the people that were scanned were reading. And you may find specific zones in the brain where you that are activated when you have concepts like remarried, husbands, father, son, sister, wife, brother. So is this the family area? No? That's a nice concept. This, I think you have not known. Family area, no. Jennifer Aniston neuron, yes, but family area, okay. So, well, family, another problem, no? And this is very interesting. You can explore the different zones, and you may have heard also of the publication of these recent papers where this information is correlated with the help of neural networks of transformers to know what the person is thinking if you have it scanned. No, so is this real? In principle, yes. But this doesn't say that we have a neuron with Jennifer Aniston. So from the machine learning perspective, about the problem, what can we say? Well, when I asked our best experts in my center about what they thought about memory, they told me, okay, you have to read Hoffield, and that memory basically in neural networks is an emerging property that comes from the scheme, and this you know very well, okay? This is a well-known view. And so the information is basically the network structure and the weights, and it's reflected in some way there. While computing, you have digital variables in the memory of the computing system, and you don't worry about engrams. Nobody talks about engrams in computing, or at least to my knowledge, not much. So what I did is I started to consider the simplest idea for storing an information, and I took the autoencoder. I assume that all of you know about autoencoders? No? Autoencoder is a simple neural network, well, relatively simple. It was not even a neural network, but the scheme is the following. You have a space with a dimension, large dimension, and through a neural network or through any other projection, you project into a smaller space with a few nodes only, and then you decode from this with another network or with the same network, with the decoder, and you train this latent space here to be as significant of the key information in the input information if you are able to decode this output so close to the input that it makes sense, it matches, okay? So what you do to train this as a neural network, you minimize the difference between the input and the output and try to make a latent space that makes sense, okay? Of course, this point of makes sense is not obvious at all. You may try different network architectures, different latent spaces, etc. So the idea is, okay, let's imagine that our brain is able to operate as a neural network, that we know more or less that it is, and generates latent space, so the images are encoded. So Jennifer Aniston would be a vector or a point in this latent space. And if we have another encoder for text that encodes the text Jennifer Aniston, that is even simpler than for images, then you have 2 points in 2 different latent space, and you just associate them with a neuron. And then this neuron that associates these points is the Jennifer Aniston neuron. It's easy, no? It makes sense, no? Or not? Well, the good point is that computationally it's trivial because you create a non-SQL database and link the different encoders, the points in the encoders to this, and you have already an engrammer, okay? So that's the point that I I want to do, but I have not done because I'm not so good on NoSQL databases. Okay, so this is an hypothesis that's quite naive, to say it softly, but as I said before, our object is not understanding our brain, but to find inspiration to explore new ideas in computing. And it has to be said that when you read the literature, there are many, many ideas exploring also naive ideas, so why not? Here is the scheme. How do you store Jennifer Aniston in your brain? Okay, Jennifer Aniston comes to your retina, goes to the encoder layers that are in the visual cortex, and then it goes to latent space for images, okay? And you have here then a memory index for images. And then you decode and you get an information about, you get an image about Jennifer Aniston, and of course, maybe as realistic as this decoder and this latent space provides, okay? But think about this. You are probably sure that if you see an image of Jennifer Aniston, you will be able to say if she is or not, no? It's not so easy to be confused with an image of a popular person. Even if it is distorted, even if it is a drawing, no? So to some point, but you see, if I ask you, okay, make a painting, make a drawing, it will not go very well, no? It's not the same, no? So it's likely that what you decode here is not the same that you encode here, the same information, okay? But it's enough to recover only this image, okay? This makes sense. Yes, this makes sense because, of course, this ability is the critical one for us to recognize something, not to reproduce the image, is to recognize, to predict that this person is this person and not another person, okay? Then you need a concept layer that basically, not here, but you have another textual perception layer, okay, where it's quite easy. You encode the Jennifer Aniston text, okay, and then you need to associate both the image and the text in another concept layer, and this is where you have a neuron that has encoded all your knowledge about Jennifer Aniston, okay? No, not okay. You cannot say okay. This way we will lose Jennifer Aniston each 2 weeks, no, or so, no, because all the synaptic connections are removed or changed, no. Do you have only one Jennifer Aniston, or you have at least 3 Jennifer Aniston neurons? It depends, no. So,
why you have to associate concept to a noun rather than say a combination of several? Why it has to be just one element rather than? Why it has to be one element rather than a specific combination of a network of eigenvectors or whatever, of a combination of several?
I don't know. Let me see the next slides, because maybe the next slide answers partly to you. So you need to integrate in this concept layer, you need to integrate everything about Jennifer Aniston. And that is one of the problems, okay? When I told these ideas to my young colleagues in my center, they told you, okay, you are wrong, totally wrong. You don't have any idea about encoders, and not even to say autoencoders, but in an autoencoder, an encoder, the node with one image of Jennifer Aniston is not close to the point with another image of Jennifer Aniston, okay? So this doesn't work, okay? But even more, to associate all these things, well, I don't know. What I can tell you is that the computing people, Meta, okay, are working on how to bind all the information. And they say, for example, that images are a perfect way to create a nucleus around to organize all the information about concepts, okay? So I don't have a better answer by now, but I have another trick in the next slides, okay? So this is the point I was saying, okay? So how to identify and associate a new image? Now there comes a second image of Jennifer Aniston, and this image are not close at all, okay? So the point is that you need to point to the same concept layer. And this is trivial if these points in the coding layer are the same, but in principle, they are not the same, okay? Because this coding layer tries to decode the image, so it is closer to this. So what does it mean? It means that the encoder, a simple encoder, cannot work with images. It needs to be deeper than a simple encoder for images. And the point is, do we have such encoder in our brain? And I was quite discouraged, but the answer is yes. The answer is that in experiments, in a paper published in 2017 by these two persons from Caltech, they analyzed how the macaques are able to recognize images of persons using only 50 plus 50 neurons, 100 neurons. So this means that with 100 neurons, you can encode all the faces that you see, at least macaques, persons are not so. So I was quite puzzled by this paper, but not so much puzzled when I read also this paper that's older of FaceNet, but maybe many of you know this FaceNet paper of Google, where they claim and they prove that they can encode any face and recognize any face with an embedded space of 128 dimensions. This means that any space of 128 dimensions is enough to code all the faces. Nice. And not only that, the point is then, if you combine this with the axons I told you before, in fact, what you need is 128 axons. Neurons, okay, but to me the key is axons. Why axons? Because with these axons around the length of the axon, you can make this, think of the axon as the axis of a dimension, okay? So you just need 120 axons and then connect them. And that's it, because this is in a space of 128 dimensions, no? Well, first of all, because 128 is 2 to the okay. But they also said in that paper, they tried many, okay? And they said, okay, we can reduce up to 128, but the other interesting point is, and this can be done with a quantized neural network. So this means you don't need real values for the values in that space, but you just can have a byte, okay? So this makes a lot of sense to make the analogy and to convince you that it may be true that our brains, one way or other, encode Jennifer Aniston face from any way in one point in a 128 dimensional space, okay? So I convinced you, no? No. Well, Maxi would be quite discouraged if you say yes, I think. Okay, so the problem is, even if we believe all these fantasies, we don't know how to build an engram in our brain, because I have said that I can build an autoencoder in a computer, but it's not so easy to build an autoencoder in a brain. And this is the problem where I am stuck. Well, one of the problems. Basically that we need to some way make this work. And in an autoencoder in a computer, this takes a lot of iterations, a lot of energy, and we don't know how to do this in a brain or with cells, okay? So some ideas are the intraneurons, or interneurons, and the role of inhibitory, that, you know, recurrent networks, that's quite an interesting idea. But then there's also another point that is more philosophical, and it's the point of why does a neuron want to compare with another neuron, no? Is there any reason? So in an autoencoder, what we compare is the distance, either likelihood on the information or directly a Euclidean measure, but this doesn't work in biology. So what is the real? So it should be either with inhibitory, is relatively tricky, but simple. You just make that inhibitory compensate the excitatory, and then one way you can reach a stationary state, maybe. But anyway, so what do specialists in artificial intelligence proposed, and this is Hinton and colleagues, and Hinton is and Lily Crabb also is, well, they are the best people. So what they proposed is to have some way of feedback so they can mimic what the neural networks do, computational neural networks, but this idea was discarded after 1 year more or less, that they can reconnect back, okay? So you see here interneurons and the reconnection back, so they can try to redo what they do computationally in biology, and this doesn't work. They have other ideas, but let's say, and that's one of the problems, that the best AI specialists are not so good neuroscientist specialists in many cases, even if they have Google behind them. Okay, so how do we go? Well, now there comes more non-science research. It is research, but it's not very scientific. So what I thought is, okay, what we have to understand, and as I will say, I will not provide any answer, and most of these are speculations. So we need many problems that are really not solved. So evolutionary understanding of how neurons work. So as I said, I tried to review the escape circuit of the C. elegans and found that it's basically molecular. I tried to understand Drosophila and also the circuits that are quite widely studied in robotics, because well, flying is an interesting movement, no? And I found also, I am still reading, it's not so easy to find some answers. I had the idea that as neuron cells come evolutionary in principle from epithelial and muscle cells, we may have there the answer. So why neurons react, muscle cells have a reaction mechanism, and you may even find an inhibitory mechanism. You know that when you move a muscle, there's another inhibitory neuron stopping that you move the opposite muscle to make the movement. No? Things like this, okay? I found yet nothing. Analyzing microcircuits. Well, you are the experts on networks of spiking coupled neurons, no? And I still have the very fundamental question if this is the real key point, the Hamiltonian of the spiking neural network, or if it is just a reflection of what happens molecularly. Well, maybe you can answer in the discussion. The dendritic computing, that's another possibility, okay, that is also discussed. How much of this is done at the dendritic level? And then there's, of course, and this is really even more speculative, to consider neurons as kind of spin glass systems with a spontaneous ordering and possible quantum effects, okay? Not only the Penrose hypothesis of the tubaling, I think is the name, but well. And then, of course, you can explore different types of engrams in different brain areas. The index model in hippocampus, the conditional learning models in amygdala. These models of amygdala, I think, have been very influential to neuroscientists, and many neuroscientists think that is action-reaction, basically, and that's one of the problems to understand. But it's clear that it has a role, and there, well, the neocortical circuit models of this. I have 4 points that we need to understand. The dynamic model of the learning process, because after all, when you talk about engrams and memory, you're talking about learning, because if you don't learn, you will not have memory. So computationally, we know how to learn. We have a clock, the computing clock, we have steps, we have a converging algorithm. Neurologically, in addition to the external stimuli from our senses, we have possible internal global clocks that, as I said, is not obvious. How the synchronization process happens, what is the temporal, does the temporal shape of signals critically influence the process or no? Metabolically, we said that the brain is very much efficient, but is it really possible that it can do all these things with the spikes, for example? And globally, do we understand anything about learning times? Is it a one-shot process or is it iterative? No? And we know that it is a one-shot process. but maybe on top of previously iterated processes. It's not obvious at all. Okay, so I think I will skip more or less this point. The point is association. The point if consciousness is required to make this association or not. That is not a trivial point also. It's just association or it's just fine-tuning. Well, this has to do with the refinement learning. And then the temporal arrow. What is the temporal arrow? It's just a neuron that drives the temporal arrow, or is just and then we can go to transformer or similar networks or what? So I will finish just reflecting about this example that I told you before, that is the C. elegans and the trigger alarm. So at some point in evolution, trigger alarm was a key point for surviving. Okay, so this is, to me, there has to be a code, and this code, as I said before, is action-reaction for many biologists directly. But did this simple code of action-reaction and then triggering escape on our motor system would give rise to a code that was able to activate this alarm faster, and then this evolved to memory? I don't know. So at the end of the story, what guides really learning? And what guides learning is something that you also know quite well in principle, and is likely predictive coding, understanding what is going to happen next. Okay, so how do we go there? In an autoencoder, it's clear that the autoencoder is going to make predictions because it matches something that you see now with something that you have seen. Okay. It is quite well known that some basic learning circuits are genetically imprinted by evolution. Okay, and then how do we get to improve these learning circuits that were genetically imprinted? Well, we don't know, but we know that it happens. Okay, we know that when we are born, we have some genetically imprinted circuits and some predisposition, but we know also that the brain is very plastic, and then you can reorient. Okay, so what we are exploring in our team, we are right now with the role of inhibitory connections in prediction, in particular with the recurrent circuits, the latent space organization in autoencoders. You asked before about the concepts. So the concept is something abstract that you may think is something hierarchically design, and how this is organized in latent space is a hot topic also in computing. Zero shot, one shot, few shot learning, and then, as I said before, building an engrammer application. That is basically an application that will run in your mobile phone, and that is very useful for all people, because when you see somebody, you will check here who is that people, and it's something that is not so obvious. Okay, thank you very much. That's all.
Thank you very much, Jesus. Very interesting and provocative talk. Any question, please? Comments? David.
I could understand this representation in this 128-dimensional space. So in a computer, there is a physical system associated to a memory.
Yes.
Then what is the physical system in the brain? Because I mean, you discuss these axons, which go through. But
the point is, if there's well, the point is that you think that 128-dimensional space is something very exotic. But it is not, because you have one axon here, and this axon has connections each 20 microns, and it has 20 millimeters. You have here up to 1,000 points, and this is just one dimension. Another axon that goes this way, it does not have to go that way. It can go in the same direction. Here you have another dimension, and another. And then simply a point is where the dendrites connect to these axons. Well, I found it too simple when I thought about that, but in principle it's true. The point was that they said the Google team, by the way, how the Google team make this because it's not trivial, it's not an autoencode. Eh? Well, the Google, well, it is, but it is not. They minimize, they took 100,000 of faces, okay? And this is a supervised learning, so they know which face is which person. And they had like, I don't know, 8 or 10 images of the same person. So they optimized the network with triplets. The triplet was an image, an image of the same person, and an image of another person. So their network should optimize that it links to the same image and not to another person. And with this, and with the infinite power of Google, they made this, and then they refined, and they found that with 128 dimensions, they were able to represent all the faces. That is really impressive. Eh?
I think that what you do is you create a neural network with 128 nodes, and with that neural network, you can recognize any face.
Yeah. That basically says that your face, even if yes. No. Of the same person. Don't forget this. It is, yeah, but it is not a continuous. That's the answer. It is a continuous theoretically, but of course we are 8,000 million people, no? Yes. So yes, you put 8,000 million people in this space. That's it. No, it was much less.
It can generalize. Well, but maybe it can generate faces that do not exist, like they are doing now. They can generate faces that do not exist, and you cannot recognize. That could be a continuous in that case. Yeah.
But you don't really need to cover all the cases. You just need to cover what happens in real life. So yeah, apparently, in 128. Well, this is what they found they could do with the FaceNet. I don't know, one database that has this. But I think it's likely that it is okay for real life, that you don't need too many. And I was not convinced either, but when I saw that the macaques can do what 100 neurons, then it's interesting. Eh?
But the software that, for instance, is used for image recognition, that the software that is used for image recognition, for person recognition, which is implemented in many places, how many parameters do they effectively use? I mean, they basically categorize the image of a person based on the distance between the eyes, the separation between the eyes and the mouth, and all these sort of things. And they recognize the person with a finite amount of characteristics. So if this is, I mean, if this 128 space parameters is the one available, they shouldn't need more than 128 effective parameters somehow, no?
No. Well, you call it parameters.
Whatever,
but it's very abstract. In fact, in the paper, I have the
variables. I have 28 variables. Whatever.
The paper shows how the different layers of the neural network goes with these images, where you see what the neural network recognizes that layer and combines convolutional neural network. So,
but how many items do you use this image recognition? Because that is somehow
the current that is used, I don't know.
How it is done, because that's effective.
Yeah, it is. But this FaceNet is the 2015, so it's already 8 years old. And in fact, I think it's implemented now in mobile phones. Not that, but other software to recognize faces.
Javier.
So
The expert on creation polish.
Also, regarding that, do you know the size in pixels of the images which they used to in the
we tried with different images, and the degradation was when it was not working well when they reduced significantly the number of pixels. But I have the paper here, as I said, not here, but in the computer. well, maybe it will take me some, but I will find it because it's just the FaceNet. But you see, and here it is. The point is that I have to say also an important problem. There are so many papers, in particular in archive, that sometimes are published, other times they are not publishing, and in particular large companies. Yeah, but when I found that FaceNet had, I don't know if it is 10,000 citations or so, no? So this must be an important paper. I have never heard of it. So here there are examples, and here is how they made the learning, the anchor with the positive and negative learning, and here is the table, the layers. Let me see here. It's a table, okay, of the image quality. Okay, so the dimensions. There was not significant. Well, they were not perfect, by the way. They were 88%, but well, I am sure now that it's improved. With 64, it was almost the same. So, but with the increase was not so successful of the dimensions, okay, but there are maybe other problems of computing. And this is sensitive to image quality. That is your question, okay. And here it says that the performance drop is very small for face thumbnails down to a size of 120 to 120, okay. And the network was trained on 220 times 220.
Because at the end, this is limited to the human level face recognition. I mean, in the data set, you label the images according to a person's. So if you put two persons that are as similar between them that I cannot distinguish them,
I think they are
the limit.
Are you sure they are labeled according to the person or according to the identity of the image?
Yeah, I haven't checked it.
Yeah, I don't know. Because it can be the identity of the person simply.
Yeah. Thanks.
I became lost in with the idea of the network connecting the different encoder representation. You have one picture of Jennifer Aniston and then another one. Why I don't know what is the what is the why is a neuron? Why it should be a network? Should be why there should be something connected?
The answer is of course speculative. Is neurons that spike together, link together. So the famous. So the idea is that, okay, you see the text, Jennifer Aniston, you see the image, Jennifer Aniston, and if both neurons, not the neurons, but the points spike activate at the same time, then the neuron of the concept will connect because it will spike together. I thought about this, that this was crazy, but there are people that are using that. Yeah. So, well, let's see, I would like to see it. But what is true is what the paper of Quiroga says, that you can associate concepts that you see at the same time, but this doesn't mean that you do it this way.
Well, I see many ways of doing it. I don't know which of them work. I remember the papers by Claudio and others in which they mentioned that perhaps is the oscillation rhythm. So the things that synchronize together are the ones that build the concept. This is another way.
That you have to have the concept in resonating in one node. But the problem for me with the resonation is the energy. No, it's not that it is not efficient. It's that I don't know the calculus of the energy that you need to maintain this electrical activity for that. I think to me it's too much.
The brain consumption is about 20 watts. It's very low.
20 watts, yes.
20, very low.
So okay, you can, and then that's the point of the quantum. No, we know that it's impossible to have quantum effects, but of course you are talking about 20 nanometers. And but here you see, this is 20 microns. So the connection here is 20 nanometers, and then the space to the next connection is 20 microns. 20 microns or 2 microns? 2 microns. Yeah, that's it. So you cannot propagate in principle a quantum effect that way, because otherwise it would be very nice to have an annealing and that's it, I learn, okay? But I think it's not possible. Anyway, even neuroscientists are exploring this hypothesis of Penrose of the tubulina, that is tubulin, that is in the cytoskeleton of the neurons. Because I said that we don't know how the connections are done, and that's a mystery. Why do you join to this or to that, no? And the excitability is not obvious, and this coupling that may be molecular, of course, it implies at some point that there's a small movement or a small attraction. So maybe the cytoskeleton is involved, and then this is what they are saying makes sense, that maybe some effect of the tubulin and the structure in the tubulin and all that. I don't know, but well, too much of speculations. Yes.
Any other question? Yes, Dimitri.
Sorry, I haven't understood exactly a part of the algorithm. So we have the autoencoder, and then we have the other the other space which has the other layer, which has the Jennifer Aniston cells. But if we could see the picture, it would be nice.
So the point is, we don't know how it works. I know how to do it in a computer. I mean, I know how to do this. As I said, what is it? Here.
Yeah.
Here. So I know.
So in the computer you have
and this is the
two
right
networks,
one, no?
right?
Hmm?
You have two parallel networks. You have
Yes,
the autoencoder, and then you have to read the latent space of the autoencoder in order to make a classification task, which is the Jennifer Aniston cell, right?
Yeah.
Because I see that there are two ideas. The one is to decrease the dimensionality, and then the Jennifer Aniston cells is a classification,
An
right?
index. Yes. And the point is that you associate in this index, you associate the different perceptions of Jennifer Aniston. And the point is, in a NoSQL database, I just know Jennifer Aniston and then write, okay, a link to an encoder, another to an encoder. The database could accept a text, could accept a sound, could accept an image, and automatically decode it, encode it, find the point, and then associate if it has been trained. Okay. So the point is, you just take all. You take first an autoencoder that is with 128 dimensions, okay, to learn faces, okay, and it has been already trained to learn faces, okay, and then you bring the first image of Jennifer Aniston, okay. This creates a point in the latent space, yes. Okay, and you put this with the tag Jennifer code, okay, in your database, okay. Then there comes a second image of Jennifer Aniston. Automatically, then the autoencoder will have a point in the same place in the latent space. Why? Because we know this is possible because of the classification of FaceNet, okay? Then, so the images are there. Then there comes a text saying, or an audio saying Jennifer Aniston. Okay, you encode it, and there's another point in another encoder that is there. And you link only when you see at the same time the image and the text, or when someone tells you this is Jennifer Aniston. So this link is the one, the association is done by simultaneous perception.
But I'm sorry, in the same autoencoder, you said that when we put two different images of Jennifer Aniston, then in the latent space, we're going to have two different
That
points.
was until I learned that FaceNet is able to do it in the same point.
Okay.
So now I do it in the same point. I don't know how, but I mean, I can do it with this encoder that is trained with triplets, okay? But I cannot do it with my brain. With something that works in my brain. In my brain, I don't have the way to, okay, now I see an image, I check with all the images people I know, and I discard, you see, you do another way. I don't know how. But it is possible. So the good point of learning this FaceNet result is that in principle, with 128 dimensions, you could store all the images of the people, or the faces of the people, sorry. Then there's another point. How do you find the face if it is in the image? But there's another area of the art to just find the, this, no? So it's another.
Okay.
Well, any other question?
So everybody convinced that Jennifer Aniston neuron is in your brains now because you have seen the image.
Now, yes, not before.
Yeah, that's a neuron to be removed as soon as possible.
Thank you very much, Jesus.
Okay. Thank you. Muy bien.
Es entretenido.
Sí.
El engram es el protocolo o el layer de los conceptos.
No, es todo. O sea, el engram es todo lo que has grabado como pesos de la red en otras capas cerebrales, eso mismo. Sí.
¿Tú te acuerdas de algo?
Más los puntos, pero es todo. Bueno, eso es por definición. No sé si quieres esa definición, bien. Si quieres otra cosa. Yes.
I think, can you use this kind of network also to recognize images that are distorted in some way?
In principle, yes, but for that, you have to train it.
Okay.
So if your brain has been trained to do that, it may do that.
Yeah, because we have a problem in Bologna, I come from Bologna, that was concerned with tumor cells, for example, which you take the cells, you put the cells in vitro, or every time that you, for example, take the cell, there's some distortion of the cell, and you will be able to recognize it again in the future. So
In
this kind of
principle, it is possible, but have in mind